Hello students, welcome to Learner's Planet. Now, today, again we are starting with the class of biology. The topic we are going to continue is microorganisms. Microorganisms, we are studying it since long, and what it means is the minute little organisms which are present in our surrounding, but they could not be seen by the naked eyes. Such organisms could be our friends or they can be our foe or the enemies. Now, we can say that these organisms are having uses as well as they can cause harm to our life. That's what we had started in the previous session. We started by studying the uses of microorganisms, and on the very first page we had studied that these uses could be of various kinds, like they could be domestic, they could be commercial, medicinal or environmental. In all these fields, these microorganisms play a very important role. Of course, right now we are studying it on the positive note only. In previous session, we had completed with the domestic and commercial uses of the microorganisms. Now, let us continue with the medicinal uses of these microorganisms. So these microorganisms, they are very important, and if we are saying that they are having medicinal importance, then what medicinal importance it could be? First of all, we must understand one basic thing. What are these medicines? For what purpose they are being used? So basically, medicinal could be some drugs or the chemical preparations which are used for the treatment of the diseases or for the prevention of these particular diseases. Now, when we are saying that these organisms are having medicinal value, then they can provide us advantage in two basic forms. That means the microorganisms helps in the preparations of some medical products which are being used for the treatment as well as for the prevention. What those things are? They are in the form of antibiotics and in the form of vaccines. The other chemical preparations, they could be synthetic or they are some artificial chemical products. Now those particular substances which are produced with the help of microorganisms and provides medicinal advantage to the person, those can fall in two categories, that is antibiotics and the vaccine. So before we study the details of these two topics or these two products, we must understand that when these products are actually used. So antibiotics, they are used for the treatment. What is treatment? Treatment is provision of certain chemical drugs or the substances so as to kill or inhibit the organism which is causing disease in our body. So in short, we can say that the antibiotics are given, they are prescribed when we are already having the disease. Whereas the vaccine, it is actually given before the disease has taken place, before the disease has occurred. That means it is used for prevention against various disease-causing agents. So this is the basic difference between the antibiotic and the vaccine. Antibiotics, they are given when the disease has already occurred, so it is called as a treatment. Vaccines, they are taken as a precautionary agent, so they help in preventing against the diseases. Now let us see how they could be available in the market. Generally, antibiotics are available in the form of capsules, or they can be available in the form of certain tablets or pills, or for the small kids, infants, those who cannot intake such large tablets or this capsules, for them these are available as a suspension, the powdered form, which is used for making the solution. And then in that solution form, it is used for the small children. Vaccine, they are generally available in the form of solutions, and these are injected in the body. There is only one vaccine which is taken orally. What is that? It is against polio virus. Whereas most of the antibiotics are taken orally, but if the severity is so high, symptoms of the disease are so high, then in that case it can be given locally. That means it could be given by injections. Directly it is injected into the blood, or in some cases it could be used as ointment, or if it has to be used at the surface of the body. So these are available in different forms, and they are used accordingly in which form it is available. Let us now understand what these antibiotics are, how they are used, how they are produced. So these antibiotics, these are basically the chemical substances. These substances, although they are chemical, but they are not produced artificially. Rather, these are the substances which are produced by some microorganisms, the living microorganisms. What is the property of these antibiotics? They are produced by living organisms, and they can kill or they can stop the growth of other disease-causing microorganisms. That means what? These chemical preparations or these chemical substances, they are produced by the living organism which do not harm itself. Rather, it can inhibit the growth of other biological agents present in their surrounding, either by inhibiting their growth or by directly killing them. So we are saying that these are produced by the living organisms. So if now consider, we said that these living organisms are producing the antibiotics, but they are killing the other organisms. So how is it possible that it can kill the other organism? It can be highly toxic for other organisms but do not cause any harm to itself. So this is the special property of such toxins or such chemical substances produced by such microorganisms. You can imagine how is it possible? It is possible because of the reaction between the substance produced and the reactant present over the another organism. For example, let us say a particular substance is produced by a particular organism, but it is having compatibility with that organism. The design is such that it cannot affect the same organism. Rather, if any other organism is present and another reactant or the substrate is present over there, these two can react. And if such type of reaction takes place, then definitely the normal biochemical reaction of other organism is going to be stopped. And this is why the organism is not able to grow. This can be more clear if we quote some example here. Let us say there is an antibiotic penicillin. You all must have heard about this particular word. It was the very first antibiotic to be discovered. Say penicillin is there and it is produced by the fungi, penicillium. Now this particular penicillin, it do not react with the fungi components, but it is having more compatibility, it is having more affinity for the cell wall product of bacteria. So in case bacteria comes in contact with this penicillin, then what will happen? The bacterial cell wall is not able to be synthesized. Cell wall is containing some glycoproteins over there. So if penicillin is present, bacteria is not able to produce the component of the cell wall. If cell wall is not produced or it is not manufactured, it is not generated, what will happen? The bacterial cycle is going to be stopped. The growth will be inhibited. They are not able to inhibit, they are not able to grow further. And this is why the organism or the penicillin is controlling the growth of bacteria. So this is how the antibiotic is actually functioning. Now you imagine, you can think that even the fungi is having the cell wall. Then why its cell wall is not affected by the antibiotic? The reason is the chemical composition. Chemical composition of both these organisms, that is fungi and the bacteria, is different from each other. So the component present in the fungal cell wall is not able to react with the penicillin. Rather, it is having affinity for the bacterial cell wall components. This is how the antibiotics are functioning. Now, the very first antibiotic was actually discovered in 1929 by Sir Alexander Fleming. He was the first person to understand, to discover this particular class of chemical substance. Which antibiotic was discovered for the first time? It was penicillin, and the source was fungi. Which fungi? It was penicillium, and the species or the strain was notatum. This penicillium was producing the penicillin. Now, can you think of this thing, how he had discovered this particular antibiotic? Actually, he was not concentrating towards this particular task. Rather, he was working in his lab over a bacterial culture. And meanwhile, he observed that some of his cultured plates, where these bacteria are grown actually in the lab, it is in the Petri plate. So in some of the plates, he found that the bacterial growth was inhibited. Some new colony, green colored colony was observed in these plates. And the plates which were having these green colored colony, other than the one which he was culturing. So this colony actually inhibited the growth of bacteria in that particular zone where it was present. And also it kills the other existing colonies of bacteria also. So he started working on this particular new green colony over there. And his observation was that there was some chemical substance produced by this particular green colony. And that substance does not allow the other organism to grow in its surrounding. Not only this much, it also kills the existing bacterial species over there. So further, he identified that particular green colony as Penicillium notatum, and the product which was produced by this was known as penicillin. So this is how the first antibiotic was produced. These antibiotics available in the market are streptomycin and tetracycline. These are the substances or the antibiotics which are produced by the bacterial species, that is streptomyces. Now many other brands are also available which are natural as well as synthetic also. That means the modified form of such type of antibiotic. The examples are more commonly like cefixime, like erythromycin and so on. When you go visit a doctor, you can find out they are providing us with so many different range of antibiotics so as to treat our diseases. Now, let us understand the next part of this medicinal use, that is the use of vaccines. Vaccines, as we had already said, that these are given as a precautionary material. They are injected as a precautionary or preventatory measure in our body. So, basically what these vaccines are? These are the medical preparations which are injected in our body so as to prevent us against the diseases. Now, this is what we are saying since beginning. What we actually mean by this medical preparation is that these vaccines are nothing but they are the solutions. Solution, you all know what we mean by solution? It's a mixture of different solvent and the solute. And we are saying it is a dilute solution. That means it is not so concentrate. So these dilute solutions, they contain weakened or dead microbes. What we mean by these weakened or dead microbes? What we mean by this here is microbes which are pathogenic. That means some organisms or some microbes can cause disease in our body. Now such microbes, they are modified. To prepare the vaccine, we need to modify these pathogenic microorganisms. Now they could be either dead or they could be weakened. Now what we mean by this weakened or dead is that we have converted these microbes into non-pathogenic form. Now these non-pathogenic form of organisms are introduced in a healthy body, in a body in which the infection has not taken place yet. Once these non-pathogenic microbes or the strains of those pathogenic organisms is injected in the healthy body, then it is going to activate our immune system. And what happens when the immune system is activated? In that case, our immune cells start fighting against the incoming foreign particle, which is microbe in this case. Now your system becomes activated. What will happen? On activation, they will produce the antibody against these organisms. Now you imagine this thing. When the vaccine is injected in the body, then the dead organism was actually injected. Right? So the dead organism cannot cause the disease. Rather, it has activated our immune system. So these vaccines, they are the dilute preparation, dilute solutions of weakened or dead microorganism which can activate a healthy body to fight against the organism. Actually, we said that these weakened microorganisms, they are non-pathogenic. They are not causing the disease, but they are actually playing with our immune system. To understand the action of these vaccines, let us clear it with the normal functioning of the pathogenic bacteria or the microorganism. Normally what happens? Why do we get the disease? A foreign particle enters into our body. Then what will happen? It will be recognized by the immune system. Right? Then once it is recognized, these immune cells will start growing. They will start developing and multiplying, and they will produce certain antibodies which will neutralize the effect of that pathogenic organism. So this way our body is going to fight against the incoming organism. So this way it is taking more and more time so as to respond to the incoming foreign particles. Now, not only this much happens. Besides this, the system which is, this process which is taking place due to entry of foreign particles will be kept in the memory of these immune cells. So what will happen? Again in future, if the same or the similar kind of microorganism enters our body, then these immune cells are going to get activated. And immediately, they will neutralize the effect of incoming organism. So, when the organism entered the body for the first time, your body took a little bit more time to respond to the incoming organism. Thus, the symptoms were more as compared to the second time. Second time when similar organism has entered, we were already having the memory of these organisms. So the primary response was not supposed to take place. Now this is the secondary response. Direct neutralization effect will start up. There is no need for initial recognition and the development of similar kind of cells. The cells were already generated, now they have to grow again to attack the incoming foreign particles. Now when we are saying that these weakened organisms are used in the vaccines, what they are doing? They are playing with our immune system. Why? Because they are not causing the disease. Rather, they are developing the memory against similar kind of microorganisms. So, this memory is going to provide advantage in future. How? Because if similar natural pathogenic microbe invades our body, then what will happen? It is not required to recognize it, then develop the immune cells of this kind, then develop the antibodies. Rather, this memory was already generated with the help of non-pathogenic organism. So now what is going to happen? These memory cells which will develop, they will directly attack this pathogenic bacteria and will help us for not generating the symptoms of the disease. So our body is going to be protected. Now, how this funda of vaccine came into role? How this vaccination process was actually started? So it was actually discovered by Sir Edward Jenner. Sir Edward Jenner, we can say it is quite a very long period of time. More than century has been passed when this vaccine was firstly prepared. And before this much time period, you can imagine that the conditions were so unmodernized. It was so backward. So how Sir Edward Jenner has developed this idea of vaccination? Let us understand this history. He just studied the epidemic disease in a local area. He found that there was an epidemic disease, smallpox, which regularly it was occurring in the local areas. And once it is taking place, most of the individuals which were exposed, they all were having the disease. But there was a group of some organisms, human beings, which were not affected by this particular virus, a smallpox virus. That means they did not develop the disease. So can you think which group was that? So it was the group which was actually maintaining the cattle. So now this cattle, that means cow and all, so those who were actually exposed to these cows or these organisms, they were not actually having the disease smallpox. So what he studied, he found that this group of milkmaid, they did not develop the disease because they were exposed to cattle. In cattle, a similar kind of virus is causing the disease, another disease. The virus is cowpox virus and the disease caused is cowpox. And the virus is also known as vaccinia. So he found that this virus, which was actually which has already entered into these milkmaid, it has prevented the effect of smallpox in such living organisms. So what he did, he just imagined that such exposure, prior exposure to the diluted forms of similar organism can prevent the entry or the development of the disease. Then he tried with this smallpox. What he did, he had injected the dried paste of these smallpox oils into the fresh organisms, into the healthy organisms. And then he found that during the further epidemics, these organisms did not develop the disease smallpox. So he just found the way in which the disease can be prevented if the body has been exposed to the similar kind of disease-causing agent, which is non-pathogenic. So this way he had developed the idea of vaccination. He used the term vaccine here. The reason is because this word vacca in Latin means cow, and the virus causing the disease cowpox is vaccinia. So from this vacca and vaccinia, the term vaccine has come. So this is how the process of developing vaccines and the process vaccination was started. Nowadays, the vaccines are widely given to many of the organisms. You can think of the diseases against which these particular vaccines are being given, like polio, tuberculosis, smallpox, hepatitis, BCG. So actually there are so many. Now you can think of government program. Even the government has started this immunization program. And the organism, the baby, day one when the baby has born, since then, the vaccination program is going to be started. Immunization begins from the day 1 of your life, that is 0 day itself. As it is born, within 2-3 days, the first vaccine is provided. The vaccine is BCG. So in this way, the entire program is there which is run by the government so as to eradicate such type of serious diseases like polio, tuberculosis, smallpox and hepatitis and so many more. So these programs has to be followed properly if we have to eradicate the disease entirely from the society. These are the vaccines which are given prior to infection, prior to entry of any pathogenic organism. But there are certain examples of vaccines which are given after the exposure to these pathogenic organisms. Can you think of certain examples? When the dog bites us, there are the chances of disease rabies. Rabies is caused by virus, and this virus is present in the saliva of dogs. Not all dogs. It is only those which are having can cause the disease. So if the dog bites, what we generally do? Within 24 hours, we are taking the vaccination or we are getting the vaccine, right? So what those vaccines are actually? They are the prepared antibodies. Normally, what vaccines were doing, as we have studied just now, they are activating our immune system so as to produce the antibodies as well as to generate the memory. But in this case, when the infection has already taken place or the infectant has entered the body but the symptoms have not yet generated, that means they have not started multiplying yet. So in this case, what is going to happen? We have directly injected the antibody, prepared antibody. So the virus or those pathogenic organisms, they will interact with the antibody and the effect would be neutralized. If vaccine is not given, then what will happen? Your immune system will work against it. But can you think how much time it will take? Because this is the primary response. It is exposed to it for the first time only. Memory is not present. So the response will take time. So till the time when the response is generated by the immune system, till then the symptoms will already take place or we will already have the disease. And such diseases are so rapidly takes place. They grow, these viruses, they grow rapidly in the body, even which is actually very less than the primary response of your immune system. This is why the prepared antibodies are required to be injected in such type of cases. So there are many more vaccines which are given here, given further in our entire life. And this process is really very important. So now we can summarize here that how these microorganisms are useful in the medical field also. That is in the preparation of the antibodies as well as, antibiotics as well as in the generation of the vaccine. In one way, we can provide the treatment. In other way, we can prevent ourselves from the disease. So this is all about this session here. Next, we are going to understand a study about the environmental uses of organisms. Thank you so much. We'll continue further in the next session. Have a nice day.